So, yes, good evening to all the students. We are here at our last session from the chapter Sexual Reproduction of Flowering Plants. As we know, this is the major chapter. We have learnt it by fragmenting the concepts. So far, pre-fertilization events are done, pollination is done, and abiotic, biotic pollinating agents are dealt, and here we are with double fertilization, where we have to begin with pollen pistil interaction. So, first thing you remember beta from pollen pistil interaction, keep the heading. Pollen pistil interaction. Look into the concept carefully. Okay. Whenever compatible pollen grain, as we have learned from the structure of the pollen grain, in 60% of angiosperms, how pollen grains will be dispersed? Two-celled stage. Remember, it is two-celled stage in 60% of angiosperms. Fine. What are those two cells, ma? Vegetative cell and generative cell. Vegetative cell is large, okay, vegetative cell is large with abnormally shaped nucleus, generative cell is small, which is floating on the cytoplasm of vegetative cell. So, here this two-celled stage, it will be dispersed, right ma? And we have learned from the structure of pollen grain that the outer wall is what? Exine. Exine wall layer will have small apertures or pores. Those are called as germ pores. Do germ pores have any sporopollenin thickenings? Does germ pore have any sporopollenin thickenings? Just so, here you see beta. In the exine region at the germ pores, you tell me whether sporopollenin is present or absent. Sporopollenin in germ pore region.
Germ pore region not present.
It is not
Only
present.
exine.
Yeah, in exine, complete exine, sporopollenin is present. At the apertures or germ pore region, sporopollenin is absent. Okay. Why? Whenever a compatible pollen grain lands on the stigma, okay, when lands on the stigma, what happens? At the germ pore region of the pollen grain and at the surface platform of stigma, there is a long chemical dialogue that happens, okay, and the signals of stigma have to accept this pollen grain. Once it is accepting, then pollen tube growth will start like this, okay. For example, on the stigma platform, two compatible pollen grains have landed at the same time, okay, then two pollen tubes will grow at the same time, no problem in that, okay. Now, in this pollen tube which is growing all across the style of the female reproductive organ, what happens is in the pollen tube only, the generative cell will undergo one more mitotic division. Yeah, it will undergo one more mitotic division, forming out two male gametes. Fine, it will form out what? Two male gametes. Fine, right beta? In those angiosperms where pollen grain is dispersed in three-celled condition, there two male gametes will be there from the beginning. Till here though it is clear, no? Now, if you want to see the pollen tube growth in some in vitro culture, okay, yeah, 5% of boric acid, 20% of sugar solution and some minerals like calcium, magnesium should be added, okay. So that you can see what you have to do is in vitro germination for pollen tube, in vitro germination, to see the pollen tube growth, in vitro germination, take a glass slide, dust some pollen grains here, okay, provide it with what ma? Sugar solution, 20% of boric acid and minerals like calcium, magnesium. Clear till here? Okay. You have understood how 2-celled pollen grain will come, how 3-celled, what will be their compatibility range, how it will be on the glass slide. Till here everything is clear, Nama? Yeah. So this is all about pollen pistil interaction. Now how pollen tube will enter into the ovule. Okay. So if you consider this is the female reproductive structure or a single carpal unit. Single carpal unit will have single stigma, style, yeah, and ovary. Inside ovary, ovule is arranged, right? How ovule will present in most of the angiosperm? It is inverted ovule, anatropous ovule like this. Okay. So into this ovule, if the pollen grain can come from any of the end. Okay, it can come from chelazal end, it can come through integuments, it can come through integuments, or it can come through the micropylar end like this. Okay. If it is from micropyle, what do you call it as? If it is from integuments, what do you call it as? If it is from chelazal end, what do you call it as? Okay. If it is from micropyle, you address that type of pollen tube entry as what? Porogamy. Okay, it is known as porogamy. If it is through integuments, it is addressed as mesogamy. If it is from chelazal end, yeah, it is known as chelazogamy. Good evening, Abdul. Please come. How are your exams going on? Fine, no? Yeah, sit. Have some water, sit. Relax, relax. Have some water. You have water? So if it is from most of the angiosperm, it is through micropylar only, but that is porogamy. In few it is mesogamy, I told you, no. Example you can take down as cucurbids. Most important, okay. Fine. And then from chelazogamy, example you can take as casuarina. Okay. This is about what? Pollen tube entry into ovule. Okay, pollen tube entry into ovule. Clear till here? Any doubt anywhere? So first this germination is clear to you, pollen tube entry is clear to you. Next you see here double fertilization. Now you have analyzed how two male gametes have entered into the ovule. Inside ovule what is there? Female gametophyte is there. What is the female gametophyte, ma?
Embryo sac.
Yeah, female gametophyte of the angiosperm is embryo sac. What is the male gametophyte?
Pollen.
Very good. Pollen grain is the male gametophyte. What does the term gametophyte mean?
Which carries gametes.
Yeah, which holds or carries gametes. Okay, embryo sac consists of female gamete, no? Female gamete is egg cell. So here you see, inside embryo sac, egg cell is present where? At the micropylar end, yes or no? And at the center there is polar nuclei. One male gamete which is released from the, just a sec, yeah. So one male gamete which is released from the pollen tube into the embryo sac will immediately fuse with what? Egg cell.
Egg cell. Another
One male gamete will fuse with egg cell. Egg cell ploid is haploid. Male gamete ploid is haploid, where it will really produce out what? Zygote, which is diploid. Fine, right? Next, second, one more male gamete
polar.
will fuse with polar nuclei. Polar nuclei ploid is what? Diploid. Okay, central cell ploid is diploid. Male gamete ploid is haploid, which will result in the formation of polar endospermic nucleus, whose ploid is triploid. Clear, right? So, the first product of fertilization, the first cell which is the product of fertilization is zygote. Zygote is the product of fertilization in any other division of plant kingdom. In any division of plant kingdom, what is the product of fertilization? Zygote. One more additional product of fertilization in angiosperms you see is what? Primary endospermic nucleus. Okay? Additional product that you see in the angiosperms is primary endospermic nucleus. A small question for your recap. In two-celled condition, pollen grain which is dispersed in two-celled condition, okay, where the mitotic division take place, where the formation of male gametes will take place, in which region? In which region the mitosis of generative nucleus of two-celled pollen grain will take place? Pollen tube which is growing in the style. In that region mitotic division happens and two male gametes are produced. Yes. Yeah, that applications I will tell you when we are solving questions. As of now you remember haploid male gamete, haploid female gamete, diploid here, haploid male gamete, diploid polar nuclear, triploid here. Based on the ploidy application you will get some application based questions that we will solve after the chapter. Okay? And here one more point to be delivered, that two fertilizations are happening, so it is addressed as double fertilization, and here triple fusion is happening, 3 nuclei are involved in fusion. Okay? So, it is also known as triple fusion. One more thing, in an embryo sac how many nuclei will be there beta? 8 nuclei will be there, 7 celled 8 nucleated condition, right? Of 8 nuclei, how many nuclei are involved in the double fertilization process? 3 nuclei are involved. Okay? If how many nuclei from embryo sac are involved in double fertilization, if that is your question, your answer should be 3. If at all, in double fertilization, how many nuclei are involved? Your answer should be 5, because he did not mention from embryo sac, no? Understood? Yes or not? In double fertilization, total 5 nuclei are involved. From embryo sac, 3 nuclei are involved. Okay, take down. From here only you take down. So, three nuclei from embryo sac. Overall, 5 nuclei involved in double fertilization. Clear, right? Any doubt anywhere? Clear till here beta. So, double fertilization is clear to you, no? Next, we will move on to post fertilization events. So, immediately when zygote and primary endospermic nucleus is formed, zygote will undergo sequential mitotic divisions which will help in the formation of embryo development. Zygote develops to what ma? Embryo. Zygote develops to embryo by undergoing sequential mitotic divisions. But prior to this, prior to embryo formation only, endosperm will be developed. How endosperm is developed? From primary endospermic nucleus. Primary endospermic nucleus again undergoes sequential mitotic divisions or mitosis that will be matured to what? Endosperm. So, endosperm development occurs prior to embryo development here. Two post fertilization events. Inside embryo sac what happens? Inside embryo sac, what happens is these cells will be degenerated ma. Synergids, yeah, synergids will be degenerated, Antipodals will be degenerated.
Ma'am, what's the point of
Antipodals, they are, every cell there is nutritive in function. It will help in the process of double fertilization. Antipodals are also addressed as vegetative cells. Vegetative cells. Being in the chelysal end, they will provide the enough nutrition for nucellus cell to differentiate into megaspore mother cell, and it will help in the nourishment of megaspore mother cell for undergoing the meiotic division. It will provide the nourishment for what? Functional megaspore for embryo sac formation, and after fusion also for development of this. The structural degeneration will happen. Structurally the structures will be degenerated, but that will be in nourishment or nutritive function for the development of embryo and endosperm. Okay. Antipodals are also known as what? Vegetative cells. Same the case for synergids. Synergids are also known as helper cells or cooperative cells. Helper cells or cooperative cells. Clear till here, ma? Yeah. Now you see, seed formation and fruit formation. This happens simultaneously. Two more post-fertilization events in angiosperms include seed and fruit formation. Which occur simultaneously. Clear? Easy? Okay. One question to you. When pollen tube is entering into the embryo sac, okay, how many synergids will be degenerated? One synergid will be degenerated while the entry of pollen tube, because you know pollen tube entry is guided by what, ma? Filiform apparatus, right? So when pollen tube is entering through one of the filiform apparatus, okay, filiform apparatus will be degenerated. One filiform apparatus only. As such, one synergid cell will be degenerated at the entry of pollen tube. Later, after fusion, one more synergid cell and the antipodals will be degenerated. Clear, right? Note down that point. And then we will move on to the endosperm. Okay. So endosperm development is of two types, ma. First type of development is nuclear endospermic development. Nuclear endospermic development. Second one is cellular endospermic development. So here in the nuclear endospermic development, what happens is primary endospermic nucleus will undergo sequential mitotic divisions. Whenever I address or use the term mitotic division, you have to be clear on the note, karyokinesis and cytokinesis will happen. But here what happens in this pattern of development is continuous karyokinesis will take place, beta. Continuous nuclear division will be taking place. Okay, nuclear division will happen, but cell wall formation or cellularization will take place later. Best example for this is coconut water. First complete karyokinesis will take place, later at the end cellularization will happen. That is free nuclear endospermic development. Okay, free nuclear endospermic development. Next is cellular endospermic development. Here, any doubt, Abdul? Primary endospermic nucleus will undergo karyokinesis followed by cytokinesis. Karyokinesis followed by cytokinesis. Okay. So this helps in the immediate cell wall formation. Immediate cell wall formation, which leads to immediate cellularization and forms what? Cellular endosperm. Cellular endosperm. Example is coconut white kernel that you see in coconut. Clear till here? Next you see. Here we go. So, types of the endosperms based on the ploidy, based on the stony endosperm, what type of endosperm, and based on the composition of endosperm. In few families, endosperm is absent. Okay, endosperm is absent in few families like Podostymaceae and Trapezae. Podostymaceae and Trapezae, these are the families where endosperm is absent. And few you will see stony endosperm, example in betel nut, okay, that is areca nut, and in date palm, that is Phoenix dactylifera, you will see stony endosperm. And in few plants like Oinothera, the endosperm ploidy will be diploid. Okay. And here you see nature of endosperms. In cereals, you can see starchy endosperm. Cereals like rice, maize and wheat, you can see starchy endosperm. And in cereals like maize, you will have proteinaceous endosperm, which has what layer? Aileron layer. And then castor and coconut will have oily endosperm. Ivory palm has cellulosic endosperm. Date palm will have hemicellulosic endosperm. Just take down this, I will be back in a minute. Okay, in just 1 or 2 minutes I will be back. Yeah, here we go. After this we will be solving the PYQs also, NEET PYQs, so be attentive. Fate of endosperm. So in few seeds, endosperm will not be completely consumed by embryo development beta. Okay. In few seeds, endosperm is still persistent in the seed. Okay. Such seeds you will see mostly monocotyledonous seeds. Mostly monocotyledonous seeds are endospermic seeds. Endospermic seeds are also addressed as albuminous seeds. Okay, endospermic seeds are also addressed as albuminous seeds. Endospermic or albuminous seeds. And then non-endospermic in woos, embryo development, endosperm is completely consumed. Okay. So, here these are what? Non-endospermic seeds or X-albuminous seeds. Non-endospermic or X-albuminous seeds. So, here we go. Examples of endospermic seeds will be mostly monocots like coconut, rice, wheat, maize and barley. Only dicot that is endospermic is castor, from which you will get castor oil. Okay, this is the only dicot. Same the case here you see, where endosperm is completely consumed, mostly you will see dicot seeds are what? X-albuminous or non-endospermic seeds. Only dicot which has endosperm, that is castor. Monocot which has endosperm, you can take orchid. Sorry, which does not have endosperm, orchid. Clear? Anyhow, at the types of seeds, we will recall this concept again. Just take down the examples, that is enough. Done, ma? Next you see embryo development, very important. We will get the questions on embryo development not only in NEET but also in the EAMCET. Okay? So, you have to focus on the dicot embryogeny, monocot embryogeny, which is nothing but dicot embryo development and monocot embryo development. Dicot embryo development. So, here first zygote is formed, right? What zygote will undergo? I told you, mitotic division. So, one zygote cell will undergo mitotic division, first transverse division. First transverse division will help in the formation of two cells, one is terminal cell, one is basal cell. Basal cell is at the micropylar end, terminal cell is at the chaliceal end, that is towards the antipodals. Okay. Here this basal cell will again undergo sequential mitotic divisions, like 6 to 8 mitotic divisions will happen, and this will form what? Suspensor cells. This complete region is called as what? Suspensor. The apical region of the suspensor is addressed as vesicular cell or hostorial cell. Vesicular cell or hostorial cell, and the basal cell of the suspensor is noted as hypophysis. Basal cell of the suspensor is noted as what? Hypophysis. This terminal cell will undergo transverse division and vertical divisions, which are known as anticlinal and periclinal divisions. Anticlinal and periclinal divisions. What are anticlinal divisions, beta? This type of divisions, perpendicular divisions, are known as anticlinal divisions. Parallel divisions or transverse divisions are known as what? Periclinal divisions. These divisions of the terminal cell will result in epibasal cells and hypobasal cells. Epibasal cell will give rise to, okay, plumule and cotyledons. Hypobasal cells will give rise to hypocotyle. Hypocotyle, radical. They will come from what? Hypobasal cells. From epibasal cells, what will come, beta? Plumule and cotyledons. Okay. So, first this is known as proembryo. In a proembryo, you will have short of epibasal, hypobasal cells, you will have a hypophysis, suspensor cell. Apical cell of suspensor cell is known as hostorial cell or vesicular cell. Clear, right? This proembryo will develop into a globular embryo. Globular embryo will develop to a heart-shaped embryo. Heart-shaped embryo will develop into a mature embryo. If you see in your EAMCET previous year questions, one question will be there on the stages of dicot embryogeny. First is what, ma? Proembryo, followed by globular embryo, followed by heart-shaped embryo, followed by mature embryo. So, in a mature embryo, what happens? You can see two cotyledons in a dicot, and at the apex you can see the plumule at the shoot apex. At the base you can see hypocotyle. Hypocotyle will mature to what? radical. Do you know what is epicotyle and hypocotyle? What is epicotyle? You said you know. Epicotyle and hypocotyle are nothing but they will have some primordial cells. Primordial cells are nothing but meristematic cells. Okay. Epicotyle will have meristematic cells which will divide continuously, forming out what? Plumule. Hypocotyle will also have these cells, primordial cells, which will mature to radical. At the tip it is having root cap. Okay. You know means you have to know it. Again, don't say don't know. Okay. Clear, right? Two cotyledons, plumule, hypocotyle, radical and root cap. Remember the diagrams carefully. You will get diagram based questions. Expectation is there. Previously you got diagram based questions in your NEET PYQs from morphology. We can expect from sexual reproduction of flowering plants this time. So here heart shaped embryo you will see only in dicots, but you will not see it in monocots. Okay, like that you can remember, because it is having two cotyledons. Fine, right? Any doubt here? Did you note down the stages properly? Take it down quick. Quickly take it down. Pro-embryo. Done, ma? Yeah. Next we see monocot embryonic development. Take down. Done? Abdul, make it little fast, ma. Next. Monocot embryo development. Here we go. Same the case, zygote is diploid. This will undergo one transverse division, forming out terminal cells and a basal cell. Basal cell will again undergo one more mitotic division, giving three-celled condition. One is suspensor cell, of course. Second will be middle cell, and at the base terminal cell will be there. Terminal cell is at what end? Chalazial end. Suspensor is at what end? Micropylar end. As such, the same development pattern you will see. Okay, and this will result in the formation of what? Globular embryo. Globular embryo will not further develop into heart shaped embryo, because it just has what? Single cotyledon. Okay, and these are what? Remnants of second cotyledon. Second cotyledon will not form, but the initials of the second cotyledon will be there. Those initials are addressed as what? Epiblast here. Okay, so clear? Three-celled condition, same sequential divisions will result in what? Globular embryo, and then single cotyledon. Here you see in a monocot embryo. Remember, important diagram, monocot embryo. From base to tip you see. Okay, this is called as scutellum. Scutellum is papery cotyledon. It will not provide any nutrients. Usually cotyledon and means what is? It has leaf, it is known as embryonic leaves. Cotyledons are known as embryonic leaves, which will provide nutrients to the developing seed at the time of germination. But scutellum is addressed as papery cotyledon because it itself will not provide any nutrients. It will drive the nutrients from the endosperm and it will give to the embryonal axis. Okay. At the tip of the embryonal axis you will have shoot apex. Shoot apex is covered by a protective sheet known as coleoptile. Okay. At the base you will have what? Radical, which has a protective sheet, coleorhiza. And this protective sheets, that is coleoptile and coleorhiza, you will see only in the monocot embryo, you will not see in the dicot embryo. Clear? Okay. How the scutellum is providing nutrients? By driving it out from endosperm. Clear? As it is only monocot embryo only we are discussing, there is no endosperm over here, but endosperm will be over here. If you can see monocot seed, okay, if you can see monocot seed, endosperm will be over here. From here scutellum will be driving out nutrients to the embryonal axis. Can you remember the monocot seed diagram? Here it is aileron layer, and inside which starchy endosperm will be there. This is scutellum. From scutellum region it is given in the embryo. This is driving out nutrients to the embryonal axis. Clear? As such, plumule radicle will grow, shoot development will happen, root development will happen. Clear? Take down. Monocot embryo development. In your simple terms. Clear? If embryo development is done, then seed formation and fruit formation, then previous year questions. What is that? Revise the examples. If you don't revise, it will volatile easily, Kunal, I am telling you. Done? Next you see. Seed. As we have discussed, first you have to know parts of the seed. First is what? Seed coat. Next is endosperm. Next is embryo. Embryo consists of embryonal axis and cotyledons. So here from these parts, based on the presence and absence of endosperm, we have two types of seeds. Based on number of cotyledons, we have two types of seeds. Fine, right? So first you see seed coat. If you have to observe in a dicot seed coat will have two layers. Outer layer of seed coat is known as testa and inner layer is known as tegmen. Testa and tegmen. Super, you are super knowledgeable, very good. Next is what is based on the presence or absence of endosperm, I told you already. At the time of endosperm development only we discussed. Okay, most of the dicots are non-endospermic or exalbuminous. Only dicot seed from our NCERT if we can take is castor seed, from which we can extract castor oil. Most of the monocots are endospermic seeds. Okay, few of them are non-endospermic. Best example you can take is orchid. Next is embryo. Okay, if you take a dicot seed, the embryo will have what? Embryonal axis at the center. Two cotyledons will be there. Two cotyledons. These two cotyledons will supplement the embryonal axis with nutrition, so that upper shoot will be growing, plumule will be developed to shoot, and lower root system will be developed. Okay, this is in the dicots. In monocots, how it will happen, I already told you in the monocot embryogeny. Okay, so here embryo will have what ma? Cotyledons and embryonal axis. So cotyledons, plumule, from epicotyle plumule will grow, from hypocotyle radicle will grow. Point of attachment of epicotyle and hypocotyle is cotyledons. This is the point of attachment for both of epicotyle and hypocotyle. Clear? Fine, right? Okay. Next, albuminous, exalbuminous seeds. Just take down the examples. Pea, groundnut, bean. Here for albuminous, wheat, maize, barley, castor. Took already? Next, seed viability. Here what is important, I will tell you. So seed after formation till the time of germination, it should leave, right? Both in favorable conditions it should leave, in unfavorable conditions also it should leave. Preventing all the external and internal barriers, it should be able to germinate accordingly. So the tenure or time period from seed formation to seed germination is known as seed viability. It can be for few months you will see it in oxalis. In oxalis you can see it for few months. Okay. In oxalis it is for few months. Seeds of many plant species is viable for years. For several years seeds are viable. Okay. Like for example, if you see some seeds which like Lupius arcticus seeds, okay, which are excavated means taken from the region of Arctic tundra, this seed successfully germinated after 10,000 years. This is the direct example of your NCERT we can expect. Okay. So, what is the seed viability period of Lupinus arcticus? 10,000 years. From which region it is extracted? Arctic tundra. That is one thing you have to remember. And during archaeological excavation at King Herod's palace, okay, at King Herod's palace, the Dead Sea, which is the Dead Sea age is around what? 2000 years old, okay. Date palm, whose scientific name is Phoenix dactylifera, it was discovered. How much age old? 2000 years old viable seed. 10,000 years old viable seed is from Lupinus arcticus, from Arctic tundra. Fine, right. The fruit of orchids will contain thousands of minute seeds. Parasitic plants like Orobanche and Striga, they will also produce numerous tiny seeds. Take down examples, that is it. Examples, years and regions. Quick, we need to go for fruit, then special modes of reproduction as well. Done? Good. Next is fruit. We have different types of fruits like fleshy fruits, dry fruits, true fruits, false fruits. We have dealt in more clear way when we are doing morphology. But here you remember types of fruits based on the development. We know ovule post fertilization will form what? Kunal, ovule post fertilization will form? Ovary? Fruit wall specifically. So, here if a fruit is completely developed from ovary, that is addressed as true fruit. Examples include mango, kiwi and grapes. Okay. False fruit, if any other floral part is also involved in the fruit formation along with ovary, such fruits are addressed as what beta? False fruits.
Tomato.
Yes, apple, strawberry, cashew. And few fruits doesn't have seeds. That is because of absence of fertilization. Okay. So, those fruits are called as what? Parthenocarpic fruits. Fruit formation without fertilization. Without fertilization. Parthenocarpic fruits examples, banana and seedless grapes. Diagrams important again, see strawberry and apple. We can expect in the diagram based questions. So, here you see this complete pulp that we eat of apple is what? Thalamus of the apple. Thalamus is also involved in fruit formation here. Done ma? Types of fruits. Next is special modes of reproduction. Here we have learned third type of fruit as parthenocarpic fruit, right. So, how the seedless fruits are formed? First, how apomictic seeds. What are apomictic seeds? Seeds which are formed without fertilization. Don't get confused. Apomictic seeds are the seeds which are formed without fertilization. Parthenocarpic fruits are fruits without fertilization. Okay. Here what happens is in the embryo sac, which is how many nucleated condition?
7 cell
8 nucleated condition. So, here polar nuclei ploidy will be what? Diploid, and embryo sac is present inside the ovule. Yes or no?
Yes.
Here if at all the tell me first, nucellus ploidy will be?
Nucellus triploid.
The cells which are present in the ovule?
Diploid.
Diploid. Integuments? Diploid. The megaspore mother cell, which is differentiated cell of the nucellus at micropylar region? Diploid. Nucellus, which is differentiated into megaspore mother cell. Ploidy is what? Diploid, right? Ploidy is in your mind. So, if at all without fertilization only, either any of the integument cells or nucellus cell is differentiating into egg directly. Egg will later develop into embryo. Understood? Okay. So, first process is what? Either nucellus cell or integumentary cells. will be differentiated into what, Nana? Egg. Here ploidy is what? Diploid. Then egg ploidy will be what? Diploid. So here embryo is directly developed, right? Is there any fertilization? No. Fine. Second process, listen first, listen. Second process is megaspore mother cell. Okay. Second, megaspore mother cell. Ploidy is what? Diploid. Okay. Megaspore mother cell is differentiating into egg cell directly. Okay. Or you can, in between you can add functional megaspore. Functional megaspore will be developed to diploid egg to diploid embryo. Okay. So, this first process is called as adventive embryony. Just a sec. Yeah. Take down. This first process is addressed as adventive embryony or sporophytic budding. Okay. And this is known as recurrent agamospermia. Fine, right? Any doubt anywhere? So, can I call this as asexual reproduction, but this is mimicking sexual reproduction? Can I call it like that? Yes. Apomixis is a type of asexual reproduction that mimics the sexual reproduction. This is seen in most of the poaceae members like grasses. Okay. Here from the diagram also you can see diploid egg cell is formed. Fine, right? So, apomixis is a type of asexual reproduction
Fruitless seeds.
You don't give new definitions, learn it as it is, huh? No, not encouraged. This is a type of sexual reproduction which mimics, this is a type of asexual reproduction that mimics sexual reproduction. Kunal. Fine, right? Yeah. Next. So, such seeds which are formed as a result of apomixis are known as what? Apomictic seeds. That is it. Done? Next about polyembryony. Done, only 5 minutes. Hold on. You all are upcoming doctors, right? You should be patient enough. Okay. Next, here we go with polyembryony. Okay. Just now we have learnt how embryos will be formed from nucellus, might be from integumentary cells, might be. Okay. As such, along with the nuclear embryo which is formed inside the embryo sac, okay, any other embryos are developing directly from the nucellus cells or integumentary cells. Such a phenomena will give you what? Multiple embryos in a single ovule. Yes or no? Along with the nucellar embryo, you will get embryos as a result of sporophytic budding from nucellus and integuments. As a result, multiple embryos are seen in a single ovule. Such condition is called as polyembryony, which is reported by first time by Leeuwenhoek. It is reported for the first time by Leeuwenhoek in citrus plant. Now it's questionnaire time. Drink some water, be active. Done with the chapter. Yes, let's get started with the questionnaire. Huh? First question, NEET 2025.
R is false, A is true.
8 nucleated and 7 celled. Be here, be here. R is false. The egg apparatus has 2 polar nuclei. Wrong. Egg apparatus will have how many cells?
8. Egg apparatus 1.
This is the embryo sac. Micropylar end, chelicel end. Here 3 cells will be there. 2 synergids,
1
egg cell. Together you will call it as what?
egg apparatus.
Haan. You will get bored now in your reasoning class. What about your memory here? Such a simple question. If you are having Alzheimer's, then it is difficult. See. So egg apparatus includes what, na?
2 synergids, 1 egg.
Take it down. What is the right answer?
A.
Next question you see. Here we go. Again assertion reason. See, from this chapter you will get most of assertion reason questions, most of statement based or match the following type. Types of pollinations important, see, both biotic, abiotic.
Assertion and reason are true, but reason is not correct explanation.
What is your answer, ma? See, both water and wind pollinated flowers are not very colorful because they do not produce any nectar. They are nectarless. So this is correct. The flowers produce enormous amount of pollen grains in wind and water pollinated flowers. There is no link only. Okay, but 2 points are correct. What is the right answer here?
4.
Good. Next question.
Nucellus.
Which part of the ovule stores reserve food material? Nucellus. Tell me the ploidy of nucellus.
2N.
Integuments,
2N.
placenta,
2N.
funicle.
2N.
Funicle is the
Point of attachment of placenta to into cellular.
funicle is the stalk of the ovule, where ovule will be attached to the placenta of the ovary. Learn the terminology. Next, this question. Pollen grains remains preserved as fossils due to the presence of good, because poropollenin is present in the exine, right? Next. Wind pollinated plant inflorescence showing flowers with well exposed stamens, okay, which is option 2. Good. Next question you see, ma. Can you see it properly? What is the function of tassels in corn cob? What are tassels? Huh? That things which you see like this, na, from corn. These are tassels. Your head. To disperse for the easy dispersal of pollen grains. Next. Statement based, see. Whenever statement based questions are given now, first hold on and look into whether he is asking you the to identify correct or incorrect statements. Then you go for answering the question.
Ma'am, D is wrong. Pollen grains or some hydrophytes?
How come D is wrong? Remember hypohydrophily?
Pollen grains.
Hmm. First statement you see beta. The flowers of Vallisneria are colorful and produce nectars. Vallisneria mode of pollination is abiotic, epihydrophily, right? Most of the flowers of water lily are not pollinated by water, true. Water lily and water hyacinth not pollinated by water. The most water pollinated species, pollen grains are protected by wetting, it has a mucilaginous coat. Pollen grains of some hydrophytes are long and ribbon like. Hypohydrophily, where pollen grains are ribbon like so that they can easily swim in the water. Next, in some hydrophytes the pollen grains are carried passively inside the water. What is the right answer? That's it. Next. Large, colorful, fragrant flowers. Insect pollinated flowers. Good.
Bird can also come.
No, bird will have beak, no? So you will have strong styles there. Strong style. So that it can withstand the pressure of the beak. Okay, there the features of the flower will be different for ornithophily. Not required for you. Look at this. Identify incorrect statement related to pollination. Related to pollination. Incorrect statement, see.
3.
Moths and butterflies are most dominant pollinating agents among insects. Pollination by wind is the most common, yes. Flowers produce fall odors to attract flies and beetles, correct. Pollination by water is quite rare in flowering plants, true. Yeah, and the incorrect statement is option 3. Next. Abdul, it's your turn now. You can do it. You think, okay, we'll see. What is your answer? Haploid, ploidy of synergids is haploid. Diploid, zygote. Triploid, primary endospermic nucleus. Option 2. Don't be doubtful, be confident. Huh? Next.
C. Thalamus.
Good. See, diagram based question. NEET 2022. This pulp is what? Thalamus. Next. Statement based question.
Statement 1 is wrong, statement 2 is correct.
Right? Correct. Cleistogamous flowers are invariably autogamous. Correct? Cleistogamous flowers are invariably autogamous because it will undergo bud pollination or self pollination. Statement 2. Cleistogamy is disadvantageous as there is no chance for cross pollination. True. Correct. So A is true, R is true. So sorry, statement 1 is true and statement 2 is true. Both the statements are correct. Next question. Good. See, this is simple question. Very good. Option? Next. Simple, easy questions. Don't expect such type now. B. Rosaceae and Leguminosae. Good. This one. Simple. You can answer. What are the two generations that you see in plant kingdom?
1, 3.
3, 4 is your answer. Listen first. 2 generations he asked, no? First of all, tell me what are those 2 generations? In life cycle of angiosperms. Plant kingdom we have learnt, no? You will have 2 generations in the life cycle. Good. Good. Gametophytic generation, sporophytic generation. Gametophytic generation is haploid, sporophytic generation is diploid. Simple logic. Okay. Now you apply and see.
1, 3.
Pollen grain ploid is haploid, whereas antherploid is germinated pollen grain with 2 male gametes. Pollen grain is haploid, 2 male gametes are also haploid, because male gametes are result of mitotic division from generative cell. Seed ploid is diploid, fruit ploid is clear. Next, embryo sac is haploid, which is inside the ovule. Ovule is diploid.
But option is 1, 4.
I think you need to get specs. Option B, right? 1 and 4. Okay? Clear? Easy na? We can expect this type of questions, ma. Have focus and look into it. This is a linked question from your life cycles of plant kingdom and sexual reproduction of flowering plants. Okay. Next. Hilum, good. Next.
Insect or wind? C.
Insects or wind. Yeah, good. Okay. Next. Last one. What is the fate of male gametes discharged in the synergita? One fuses with egg, one male gamete will fuse with central cell. Okay? Able to answer? This weekend you have the same thing. Once revise and you will be fine tuned. Okay? Fine, right? Yeah, that's all for today's session. Thank you.